Something happened today. The brain filed it, assessed it, and in most cases set it aside. But somewhere in the hours that follow, in the dark and the drift before sleep, something that felt gone returns, not summoned, not searched for, simply there. The hippocampus does not keep memories permanently. Research by Howard Eichenbaum and colleagues across several decades of work on the structure of memory consolidation has established that the hippocampus functions as a temporary holding site, a kind of way station where newly acquired memories are stored in rapidly accessible form, awaiting the process that will transfer their essential contents to the neocortex for long-term storage. This process does not happen during waking. It happens during sleep, when the hippocampus and the cortex enter into a coordinated conversation, a back and forth of electrical signals through which the day's experience is reviewed, processed, and either filed permanently or released. The hippocampus is not the library. It is the loading dock, open all day, and the work of sorting happens after hours. If this is the kind of thing you want to fall asleep to, please like and subscribe. Sharp wave ripples, brief, high-frequency bursts of electrical activity generated in the CA3 and CA1 regions of the hippocampus during non-REM sleep are among the most powerful and most specifically memory-relevant events in the sleeping brain. Research by György Buzaki, whose work over several decades has defined much of what is known about hippocampal oscillations and their role in memory, has established that sharp wave ripples represent the hippocampus at its most synchronized, a population of neurons firing together in a compressed, rapid burst that appears to replay the patterns of activity from previous waking experience. Studies measuring the precise timing of place cell firing during sleep have found that the sequential patterns of neural activity that occurred during navigation or learning are reproduced during sharp wave ripples at speeds approximately 10 to 20 times faster than the original experience. A rapid, compressed replay of the day, running in the dark while the conscious mind rests nearby. The sleeping brain is playing the day back, faster than it happened, and in a language the conscious mind will never hear. The discovery that the sleeping brain replays waking experience was made by Matthew Wilson and Bruce McNaughton at the University of Arizona in 1994, when they recorded the activity of place cells, neurons in the hippocampus that fire when an animal occupies specific locations, in rats during maze navigation, and then during subsequent sleep. They found that the same sequences of place cell activity that occurred during waking navigation were reproduced during slow wave sleep, with a temporal structure that preserved the ordering of the original experience. The finding established, for the first time with direct neural recording evidence, that the sleeping brain was not simply resting, but actively revisiting what the waking brain had done. Subsequent research using more sophisticated recording methods has replicated and extended this finding in multiple species, including humans, across a range of memory types: spatial, episodic, procedural, and emotional. The sleeping brain does not leave the day behind. It goes back through it quietly, while everything else is still. The replay that occurs during sharp wave ripples does not reproduce the day's experience randomly. Research on the selectivity of hippocampal replay has found that the events most likely to be replayed are those that were most behaviorally significant. The turns that led to reward, the moments of unexpected outcome, the experiences that carried the greatest emotional or informational weight. This selectivity is not a post hoc filter applied after replay occurs. It appears to reflect the tagging that happens at the moment of original encoding, when the presence of dopamine, norepinephrine, and the concurrent activity of the amygdala marks certain experiences as worth keeping, flagging them at the moment they occur for priority processing during the sleep that follows. The brain does not wait until the night to decide what matters. It decides at the moment of experience and sets the flag before the memory has even formed. Mirror selection begins the moment something happens. The night only carries out the instruction. Have you ever noticed how something you barely registered during the day comes back fully formed in the moments before sleep? Systems consolidation, the gradual transfer of memories from hippocampal temporary storage to distributed neocortical networks for long-term storage, is not a single event, but a process that unfolds over multiple nights and, for some memories, over months and years. Research using lesion studies and neuroimaging has found that recently formed memories depend heavily on the hippocampus for retrieval. Patients with hippocampal damage show severe impairment in recalling recent events while retaining access to memories formed years or decades earlier. As memories age and are repeatedly consolidated during sleep, their neural substrate shifts from hippocampus dependent to a distributed neocortical representation that no longer requires the hippocampus for retrieval. The memory that returns years later is not the same neural object as the one stored the night it was formed. It has been remade slowly and iteratively by the sleep of all the intervening nights. Every night of sleep has a hand in every memory that survives. The nightly transfer of memories from hippocampus to neocortex depends on a precise coordination between three overlapping oscillatory events: slow oscillations generated in the neocortex, sleep spindles generated in the thalamus, and sharp wave ripples generated in the hippocampus. Research by Jan Born and colleagues at the University of Tübingen, and by multiple laboratories since, has found that these three oscillations are not independent. They are nested in a specific temporal hierarchy during slow-wave sleep, with the neocortical slow oscillation orchestrating the timing of spindles, which in turn coordinate with sharp wave ripples to create precisely timed windows of hippocampal neocortical communication. The memory transfer does not happen continuously. It happens in brief, precisely timed packets during the moments when all three oscillations coincide, moments that the brain generates reliably across the night, each one a small, exact opportunity for the day to be written somewhere it can last. The memory is moved in moments that last milliseconds. The brain creates those moments hundreds of times each night. The selectivity of memory consolidation during sleep is what makes forgetting meaningful rather than simply unfortunate. Research on the function of overnight forgetting has converged on the understanding that the brain does not simply try to keep everything and fail. It actively selects what to preserve and what to release, according to criteria including emotional relevance, behavioral significance, and compatibility with existing knowledge structures. The synaptic homeostasis hypothesis, developed by Giulio Tononi and Chiara Cirelli, proposes that waking experience broadly strengthens synaptic connections, while sleep selectively weakens those that were least used and reinforces those most relevant, a process of overnight pruning that reduces the metabolic cost of the day's learning while preserving its most useful contents. Forgetting is not what happens when memory fails. It is what happens when memory succeeds. The brain does not forget by accident. It forgets by design, in the service of what it has decided to keep. The experience of lying in the dark and finding that something you had not thought of in years is suddenly present. A face, a room, a specific quality of afternoon light from a time long past reflects the particular cognitive conditions of the pre-sleep state in ways that research on memory retrieval has begun to explain. As directed attention relaxes and the executive control of the prefrontal cortex gradually withdraws from the business of managing thought, the default mode network, whose activity is associated with autobiographical memory, mental time travel, and the spontaneous retrieval of personal history, becomes relatively more active. The thoughts that arrive in this state are not randomly chosen from everything ever experienced. They reflect the network's associations. The spreading activation through autobiographical memory that occurs when the controlling hand of directed attention releases its hold on what the mind is allowed to retrieve. The mind does not wander randomly in the dark before sleep. It follows a map of what is most connected to what was on the mind. Have you ever found that a problem you could not solve during the day arranged itself differently in the drift before sleep? Research on spontaneous memory retrieval, the occurrence of memories that surface without any deliberate search, has found that these unbidden memories are not random samples from autobiographical storage. Studies examining the content, timing, and neural correlates of spontaneous memory retrieval have found that involuntary memories are more emotionally intense, more sensory specific, and more likely to involve personally significant events than voluntarily recalled memories of equivalent age. The hippocampus is consistently activated during both voluntary and involuntary retrieval, suggesting that the same fundamental mechanism of pattern completion underlies both. The difference between them is not in the machinery, but in whether the process was initiated by the conscious self or by the associative structure of memory itself, following its own connections without being asked. The memory that arrives uninvited took the same road as the one that was sought. It simply left without being told to go. Targeted memory reactivation, the experimental technique in which specific cues associated with learned material are presented during sleep to strengthen the consolidation of that material, has provided some of the clearest causal evidence that the sleeping brain is actively processing specific memories rather than consolidating everything indiscriminately. In a landmark study, participants learned to associate object images with specific locations on a computer screen while hearing a distinctive sound for each object. During subsequent slow-wave sleep, half the sounds were replayed softly. On the next morning, memory for objects whose associated sounds had been replayed during sleep was significantly better than memory for objects whose sounds had not been replayed. The sleeping brain, hearing the cue, preferentially replayed and consolidated the associated memory. The sound reached into the sleeping mind and told it what to keep. What the sleeping brain is told to remember, it remembers. The cue does not create the memory. It finds it in the dark and holds it up. New research from Cornell University, published in 2024, identified a specific mechanism by which the hippocampus clears itself overnight to prepare for the following day's learning. Azahara Oliva and colleagues found that the CA2 region of the hippocampus, a subregion whose function had been less well characterized than the heavily studied CA1 and CA3 regions, generates a distinct state during sleep in which pyramidal neurons in CA1 and CA3 are silenced, creating a brief reset of the hippocampal circuit. This reset state, generated by specific interneuron circuits regulated by CA2, appears to free up the hippocampal resources that were occupied by the previous day's encoded memories, allowing the structure to arrive at the next day's experiences with renewed encoding capacity. The mechanism explains a paradox that memory researchers had long noted. Sleep does not simply preserve what was learned. It also restores the capacity to learn again. The night does not only keep the day's memories, it makes room for what the next day will bring. The relationship between sleep and memory is not simply that sleep strengthens what was learned. Research on the timing of sleep relative to learning has found that the direction of the effect depends on when sleep occurs. Sleep immediately following learning produces the strongest consolidation effects. The memories are still in the hippocampus. The encoding-related neural patterns are still active, and the sharp wave ripple replay during subsequent slow wave sleep catches the memory at its most retrievable moment. Sleep occurring long after learning, or in the absence of any new learning, produces more general maintenance rather than targeted consolidation, and wake periods following learning, before the first sleep, produce a progressive weakening of the newly encoded memory, a forgetting that the subsequent sleep arrests but may not fully reverse. The first night of sleep after learning is the most consequential one. It is the night the memory is either moved or lost. The memory formed today is most vulnerable tonight. The brain will not get another chance at the same version of it. The compression of memory during overnight replay means that what is replayed is not a faithful reproduction of the original experience, but a distilled version. The essential structure of the sequence without all of its contextual detail. Research on the properties of hippocampal replay has found that the replayed activity patterns preserve the temporal order of the original experience and the spatial relationships between its elements, while omitting much of the incidental detail that accompanied it. This is not a limitation of the replay process. It is part of its function. The abstraction of a memory from its specific context, its gradual decontextualization across multiple nights of consolidation, is what allows it to eventually become general knowledge rather than a single context-bound episode. The memory that persists for decades is not the original experience. It is a series of increasingly refined extractions from it, each night's replay taking it a step further from what it was. What is remembered across a lifetime is not the experience. It is what the brain decided the experience meant. Research on memory and identity has found that the self is not a stable object that memory serves. It is, in a fundamental sense, constructed from what is retrieved. Studies in autobiographical memory and self-concept have found that the accessibility and emotional tone of autobiographical memories directly shapes the person's current sense of who they are, what they value, and what they expect from the future. The memories that the sleeping brain selects, consolidates, and makes most accessible are therefore not just a record of what happened. They are, iteratively and cumulatively, one of the primary mechanisms through which a self is assembled and maintained across time. The night does not simply archive the person's history. It revises the person slowly and without asking, by deciding what of that history remains most reachable. The self is built from what the brain brings back. Every night, quietly, the construction continues. The distinction between remembering and forgetting is less clear at the neural level than it appears from the experience of it. Research on memory traces and their persistence has found that many memories that appear to have been forgotten, memories that cannot be retrieved under ordinary conditions, leave a detectable neural trace that can be accessed under the right conditions of cueing, context reinstatement, or altered physiological state. Studies using optogenetic techniques in rodents and cue-based retrieval experiments in humans have found that the failure to remember is often a failure of retrieval rather than a failure of storage. The memory is present in the neural substrate but cannot be reached by the retrieval process currently available to the mind. This is why something that felt gone can return, fully formed, under the right conditions, in the right place, in the right emotional state, in the drift before sleep, when the ordinary retrieval controls have relaxed their hold. The forgotten memory was not erased. It was waiting for the right kind of dark to become visible again. What the night does to memory is not something the conscious mind participates in. It happens below awareness, in the architecture of sleeping neural circuits, while the self that would monitor and direct the process has been set aside. And yet, what emerges from the process, the memories that are strengthened, the connections that are formed, the knowledge that is deepened, and the details that are released, shapes the conscious mind that wakes in the morning in ways that will persist for years. The night works on the mind without the mind's knowledge, using tools the mind cannot see, toward ends the mind will experience as simply being who it is. The person who wakes is not quite the same person who lay down. The night has been at it again. The sleep is not time away from the mind. It is time the mind uses differently, without knowing it is being used. The brain does not treat all memories equally. Something happened today that will survive the night with more of itself intact than what happened beside it. And the brain decided which one that would be before the day was over. The amygdala, the almond-shaped structure deep in the temporal lobe most associated with fear processing, threat detection, and emotional salience, plays a critical role in determining which memories receive priority consolidation during sleep. Research on the amygdala's contribution to memory encoding has found that emotional arousal at the moment of an experience triggers the amygdala to release modulatory signals, primarily through norepinephrine and the activation of beta-adrenergic receptors in the hippocampus that effectively deepen the encoding of the accompanying memory. The memory is written more completely, with more redundancy, at a synaptic level that corresponds to its emotional weight. This happens at the moment of the experience itself, before the night has arrived. The amygdala attending every significant moment and quietly deciding whether the hippocampus should hold on harder. Emotional experiences are filed differently from the moment they occur. The night inherits a set of instructions the day already wrote. The specific mechanism by which the amygdala's emotional tagging influences overnight consolidation involves the coordinated activity of the amygdala and hippocampus during REM sleep, a sleep stage in which both structures are simultaneously and intensely active. Research using neuroimaging in sleeping participants has found that amygdala-hippocampal connectivity during REM sleep is elevated above its waking level, and that the degree of this overnight connectivity predicts how well emotional memories are retained the following morning. The two structures, which cooperated during the original emotional experience to encode the memory with heightened strength, return to each other during REM to finish the work, replaying, reinforcing, and integrating the emotional components of the experience in ways that further cement its long-term accessibility. REM sleep is not rest from emotional experience. It is where emotional experience is decided upon. The emotionally significant memory gets a second encoding during the night. Most memories only get one. Matthew Walker, the sleep researcher and neuroscientist at UC Berkeley, has articulated a framework that captures one of the most counterintuitive findings in the sleep and memory literature, that REM sleep serves not only to consolidate emotional memories, but to regulate the emotional charge they carry. His overnight therapy hypothesis, developed from research on REM sleep and emotional memory processing, proposes that REM sleep achieves two opposing functions simultaneously. It strengthens the informational content of an emotional memory, preserving what happened, while reducing the physiological and emotional reactivity attached to the memory, softening how it feels to retrieve it. The mechanism involves the suppression of norepinephrine during REM sleep. The brain's main stress neurochemical goes quiet during this stage, while the amygdala and hippocampus replay the emotional memory in a neurochemical environment that is calmer than the one in which it was encoded. The experience is revisited. The alarm attached to it is gradually turned down. See Leaves does not make you forget what happened. It changes how much it hurts to remember it. Research testing Walker's overnight therapy hypothesis has found consistent support in both behavioral and neuroimaging studies. In one key paradigm, participants were shown emotionally negative images either in the morning, remaining awake until an evening test, or in the evening, sleeping before a morning test. Those who slept before the retest showed significantly lower emotional reactivity to the images, reduced amygdala activation, and lower self-reported distress, compared to those who had remained awake for an equivalent interval. The subjective rating of the memory's emotional intensity decreased after sleep in a way it did not after equivalent waking time. The emotional content of the memory was preserved. Participants recognized and correctly identified the images equally well, but the emotional weight attached to retrieval was demonstrably lighter. Sleep had kept the fact and softened the feeling. The memory survived the night intact. The pain attached to it did not survive it equally. Sleep deprivation has the opposite effect on emotional memory, not neutrality, but amplification. Research using fMRI to measure amygdala reactivity in sleep-deprived participants has found a 60% increase in amygdala response to emotionally negative images compared to adequately rested controls, and a near-complete disconnection between the amygdala and the prefrontal cortex, which under normal conditions provides the regulatory input that contextualizes and moderates the amygdala's reactivity. The sleep-deprived brain is not simply tired, it is emotionally dysregulated in a specific, measurable, and neurologically explicable way. It has lost the overnight processing that would have reduced the alarm signal attached to the previous day's difficult material, and it has lost the prefrontal regulatory capacity that would have managed the alarm. It responds to emotional memory and to the present moment with the raw force of something that has never been tempered. A mind that did not sleep did not finish what the night was supposed to do to its hardest memories. Have you ever noticed that something upsetting in the evening feels different, smaller, or simply different in quality after a full night of sleep? The Proust phenomenon, named for the novelist Marcel Proust's description in the opening pages of In Search of Lost Time, of an entire vanished world returning through the taste of a madeleine dipped in tea, refers to the involuntary retrieval of a rich, emotionally vivid, long-dormant autobiographical memory triggered by a specific sensory cue, most reliably a smell. Research on this phenomenon, reviewed by Johan Wilander and Maria Larsson at Stockholm University, has confirmed that odor-cued autobiographical memories are older, typically dating to the first decade of life, more emotionally intense, more vivid, and more likely to feel like genuine retrieval of a lost experience than memories cued by any other sensory modality. The smell does not remind the person of something. It returns the person, briefly and with unusual completeness, to somewhere. No other sense does what smell does to a forgotten memory. It does not reconstruct the past. It reinstates it. The reason that smell is uniquely positioned to trigger this kind of retrieval lies in the anatomical structure of the olfactory pathway. The only sensory system that projects directly to the limbic system, including the amygdala and hippocampus, without first routing through the thalamic relay station that mediates all other sensory information. Research on olfactory neuroanatomy has found that olfactory information reaches the piriform cortex, entorhinal cortex, amygdala, and hippocampus within 1 or 2 synapses of the olfactory receptor neurons, meaning that a smell arrives in memory and emotional processing structures with a directness and a speed that no other sensory input matches. When a smell associated with a specific experience from decades ago is encountered again, it activates the neural circuitry of the original encoding with unusual fidelity, not as a reconstruction from fragments, but as a pattern match reaching directly into the stored representation, bypassing the degradation that other retrieval routes accumulate over time. The smell takes the shortest road. That is why it arrives at a place the other roads have long since forgotten how to reach. The hypnagogic state, the transitional window between full wakefulness and stage N1 sleep, is characterized by a specific pattern of default mode network activity that makes it one of the richest spontaneous memory retrieval environments in any 24-hour period. Research on default mode network engagement during the sleep onset transition has found that as directed attention withdraws and the prefrontal executive control of thought relaxes, default mode activity intensifies, and with it, the spontaneous activation of autobiographical memory networks that characterize this network's resting function. The thoughts that arise in the hypnagogic drift are not random. They reflect the spreading activation of the autobiographical memory network from whatever emotional or cognitive content was most active during the preceding waking period, traveling outward through associative connections that the structured thought of wakefulness does not ordinarily follow. The person who lies down, still holding something from the day, will find it in the hypnagogic drift, pulling other things out of the archive with it. What is on the mind at the edge of sleep becomes the first thread of an associative chain the mind pulls at in the dark. The default mode network, active during rest, autobiographical memory retrieval, mental time travel, and self-referential processing, is suppressed during focused, goal-directed thought and released during periods of unfocused mental activity. Research on default mode network function and autobiographical memory has established that this network is the primary neural substrate through which the mind visits its own past during unstructured moments, not only in the pre-sleep period, but during any window of reduced external demand. What makes the pre-sleep window distinctive is its duration, consistency, and depth. It is a reliably occurring period of progressive cognitive relaxation in which the default mode network is increasingly unopposed by the executive systems that normally compete with it. The memories that come in this window do not feel like retrieval. They feel like arrival, as though the mind stopped moving long enough for them to catch up. The memories were following the waking mind all day. The moment it stopped, they found it. Have you ever drifted towards sleep and found a face, a place, or a moment from long ago that you had not thought of in years, arriving as though it had simply been waiting for the quiet? Childhood memories surface disproportionately in the hypnagogic drift, For reasons that research on autobiographical memory and emotional encoding can partly explain, the earliest years of life are associated with a period of heightened neural plasticity during which experiences are encoded with greater amygdala participation. Not because early experiences are necessarily more traumatic, but because the developing limbic system is relatively more influential in shaping encoding than the as yet incomplete prefrontal regulatory systems that will later moderate it. Memories formed during this period carry a stronger emotional and sensory signature than many memories formed in adulthood, and this signature persists because the neural structures that encoded it have changed, matured in ways that make re-encoding of equivalent material in adulthood less emotionally saturated. When the pre-sleep default mode network follows its associations back through the most emotionally resonant material in autobiographical storage, childhood memories are disproportionately represented, not because they are necessarily the most significant, but because they were among the most deeply written. The oldest memories are often the most vividly kept. They were written when the pen was heaviest. The cortisol rhythm plays a specific and sometimes counterintuitive role in the overnight processing of emotional memories. Research has found that cortisol levels are not uniformly suppressed during sleep. They rise gradually across the night and peak sharply in the early morning hours before waking. This means that the REM-dominant second half of the night, during which emotional memory processing is most intense, occurs in a hormonal environment of gradually increasing cortisol. Research on cortisol and memory consolidation has found that moderate cortisol elevation can enhance the consolidation of emotionally significant memories, particularly those encoded during periods of high arousal. The early morning REM periods, the longest, most vivid, most emotionally active dreams of the night, occur in a cortisol environment that is specifically favorable to the strengthening of the emotionally tagged memories that the amygdala flagged during the preceding day. The body is not fighting the memory work. It is providing the chemistry it needs. The early morning, when cortisol is rising and dreams are most vivid, is when the emotional archive is most actively organized. The role of slow oscillations in the selective transfer of emotional memories during sleep connects the night's largest electrical events to its most personally significant contents. Research using EEG to measure the temporal relationship between slow oscillations, sleep spindles, and sharp wave ripples has found that the triple coordination of these events during slow wave sleep does not occur equally for all memory types. Emotional memories, those encoded with amygdala participation and flagged with the neurochemical signals of emotional importance, show a bias toward replay during the slow oscillation upstates that coordinate hippocampal neocortical transfer. The mechanism appears to involve the amygdala's ongoing influence on hippocampal activity even during sleep. The emotional tag written at encoding continues to shape the probability of replay during the night, ensuring that the things that mattered most are given the most resources during the transfer that makes them last. The most important memories are not simply stored more carefully, they are transferred more reliably on a schedule the amygdala set at the moment they occurred. Fear memories, memories associated with threat, pain, or intense negative emotional arousal, are processed during sleep in a way that differs in important respects from the processing of other emotional memories. Research on fear memory consolidation has found that the hippocampus amygdala complex engaged during fear encoding shows especially robust replay during REM sleep, and that disrupting REM sleep in the hours following a fear experience significantly impairs the consolidation of contextual fear memories in animal models. This finding has clinical implications for understanding post-traumatic stress. Research on PTSD and sleep has found that REM sleep disturbance, which is among the most reliable and severe sleep abnormalities in trauma survivors, may disrupt the normal overnight attenuation of fear memory's emotional charge, leaving the memory repeatedly retrieved in its original high arousal form rather than progressively softened through the overnight therapy process that normal REM sleep provides. The nightmare is not the problem. It may be the disrupted attempt at the solution. The connection between REM sleep and creative recombination of stored memories. The generation of novel associations between experiences that were encoded separately has been documented in research that distinguishes REM sleep from both slow-wave sleep and wakefulness in its capacity to bridge distant material. Research by Ulrich Wagner and colleagues, and by Penelope Lewis and others, has found that participants who slept, particularly those who entered REM sleep, before being tested on problems requiring the integration of separately learned information, showed significantly better performance than those who remained awake or were limited to slow-wave sleep. The proposed mechanism involves the particular neurochemical environment of REM sleep: low norepinephrine, high acetylcholine, which is associated with reduced inhibition of associative connections that would ordinarily be suppressed during focused, goal-directed waking cognition. REM sleep loosens the normal constraints on what the mind allows itself to connect. The dream is not confused thinking. It is associative thinking operating without the constraints that waking logic imposes. State-dependent memory, the well-documented phenomenon in which information is most easily retrieved when the retrieval conditions match the encoding conditions, has an emotional dimension that shapes what surfaces at night in ways the waking mind is rarely aware of. Research on mood-congruent memory retrieval has found that the emotional state at the time of retrieval preferentially activates memories encoded in a similar emotional state. The anxious mind tends to find anxious memories. The sad mind tends to find sad ones. The relaxed, drifting pre-sleep mind tends to find memories encoded in states of comparable ease. This is not simply a poetic observation. Research has measured the effect in terms of the relative accessibility of different memory types across different mood states and found consistent and specific biases. The emotional character of the pre-sleep state is therefore not merely a backdrop to the memories that arrive before sleep. It is one of the filters through which the archive is accessed. The mind before sleep does not find memories at random. It finds the ones that match the color of what it is already feeling. There is a quality to the return of a long-forgotten memory that is phenomenologically distinct from ordinary recall. People who experience the sudden, uninvited return of a memory that felt lost often describe it not as a feeling of remembering, not as the experience of retrieving something from a location where it was known to be stored, but as the experience of something simply being present that was not present before. Research on the neural correlates of this subjective experience has found it correlates with a pattern of activity, sometimes called the memory signal, a burst of hippocampal activity in which the partial cue reaching the hippocampus from the current context activates a complete pattern of stored activity through the mechanism of pattern completion, producing a sudden, full instantiation of the original encoding in a way that arrives too fast and too complete to feel like active search. It does not feel found. It feels like it found you. The memory was not retrieved. The memory returned. The difference, neurologically, is real. What the night does to emotional memory is not simply protective, it is transformative. Research following memories across multiple nights of sleep has found that the neural representation of an emotional memory shifts progressively over time. From a vivid, context-rich, hippocampus-dependent episode encoded with the full sensory and emotional specificity of the original experience toward a more schematic, semantically organized, neocortex-based representation that preserves the meaning of the experience with progressively less of its specific detail. The emotional core tends to survive this transformation longer than the contextual specifics. People often retain a clear sense of how something felt long after they have lost the details of when, where, and how it happened. The night is gradually extracting what the experience taught from the raw material of how the experience felt, keeping the lesson and releasing the film. Over many nights, what happened slowly becomes what it means. The night is always in the middle of that translation. Not all of what was lost was taken. Some of it was given away deliberately by a brain that knew what it was doing, that was, in fact, doing one of the most important things it does all night. Forgetting has historically been framed as memory's failure, the unwanted outcome of a system trying to preserve and falling short. Research developed across decades of neuroscience has progressively replaced this framing with a different one. Forgetting is not what happens when memory stops working. It is what happens when memory works precisely as it is supposed to. An active, regulated, and specifically targeted process by which the brain releases what is least useful in order to protect and strengthen what is most useful. The synaptic homeostasis hypothesis, developed by Giulio Tononi and Chiara Cirelli at the University of Wisconsin-Madison, proposes that waking experience broadly and non-specifically strengthens synaptic connections across the brain, encoding everything encountered, regardless of its ultimate relevance, and that sleep performs the complementary function of selectively scaling these connections back to a sustainable baseline, reinforcing the most relevant and reducing the rest. Without this overnight renormalization, the brain's synaptic resources would be exhausted within days. Forgetting, in this framework, is what makes remembering possible. The brain forgets to make room for what it has decided matters. The room is finite. The night manages it. The evidence for the synaptic homeostasis hypothesis comes from multiple directions. Research measuring synaptic strength directly, using molecular markers of synaptic size and neurotransmitter receptor density in brain tissue, has found that these markers increase across the waking day and decrease across sleep in a pattern consistent with the hypothesis's predictions. EEG research has found that the amplitude of slow waves during early sleep, when slow wave sleep is most intense, correlates with the amount of learning that occurred during the preceding waking period, as though the brain's overnight renormalization task is scaled to the magnitude of the day's synaptic strengthening. Behavioral research has found that performance on tasks requiring the isolation of the most important features of learned material, filtering signal from noise, is specifically improved by sleep in ways that cannot be explained by simple consolidation of everything learned, but can be explained by the selective preservation of the most task-relevant synaptic changes and the reduction of the rest. The slow wave at the beginning of the night is, in part, a measure of how much the day put in the brain that the night must now sort through. The active nature of overnight forgetting means that the timing of sleep relative to the forgetting process matters in ways researchers are still mapping. Research on the temporal dynamics of memory loss has found that the forgetting curve, the well-known pattern of rapid initial memory loss followed by a slower decline, first described by Hermann Ebbinghaus in the 19th century, is specifically interrupted by sleep. Material tested before sleep shows the characteristic rapid decline during the first hours after learning, but after a night of sleep, the rate of forgetting significantly slows. Sleep does not stop the forgetting. Selective synaptic downscaling continues to reduce some memories, but it consolidates the survivors of that selection into a more stable, longer-lasting form, changing the slope of the forgetting curve for everything it decided to keep. The memories that make it through the first night are, in a meaningful sense, the ones that will still be there in a decade. The night divides memories into those that will survive and those that will not. That decision, made once, tends to hold. Reconsolidation, the finding that a memory, once retrieved, does not simply return to storage unchanged, but enters a brief period of lability, during which it can be modified, strengthened, or disrupted before being restored, represents one of the most significant revisions in memory science of the past 3 decades. Research by Karim Nader and colleagues, published in Nature in 2000, demonstrated in rodents that an established fear memory, once retrieved by presenting the original conditioned stimulus, required de novo protein synthesis in the amygdala to be restored, and that blocking this synthesis after retrieval caused the memory to fade, even though it had been stable for years before retrieval. The memory was not simply read and returned. It was read, briefly disassembled, and reconstructed, and during the reconstruction, it was vulnerable. Every time a memory is retrieved, It is briefly the most fragile it has been since it was first formed. The act of remembering undoes part of what made the memory stable. For a moment, the past is open to revision. The reconsolidation window, typically estimated at approximately 6 hours following retrieval, has specific implications for the role of sleep in restabilizing retrieved memories. Research on sleep and reconsolidation has found that sleep occurring within this window following memory retrieval produces significantly more stable reconsolidation than wakefulness in the same period. The retrieved memory, in its temporarily vulnerable state, is preferentially processed during the subsequent sleep in ways that restore and often strengthen its representation. This has been proposed as a therapeutic mechanism. The vulnerability of reconsolidation, combined with the emotional attenuation of REM sleep, may provide a natural window during which difficult memories retrieved during the day are restored in a form that carries less of their original emotional charge. The night that follows a day on which old pain was revisited may be doing specific and important work, not simply rest, but the restabilization of something that was broken open during the day and needs to be closed again more carefully. What was opened during the day needs the night to be closed with less pain than it carried before. Trauma memories resist the normal overnight attenuation process in ways that research on PTSD and memory has carefully documented. The neural encoding of traumatic experience involves extreme amygdala activation, high norepinephrine and cortisol levels, and a pattern of encoding that prioritizes the central emotional core of the experience: the threat, the fear, the helplessness, at the expense of the contextual details that would normally allow the memory to be filed as a specific past event. Research has found that this encoding pattern produces memories that are simultaneously more intrusive, more likely to surface unbidden with high emotional intensity, and less coherently organized than normal autobiographical memories. The hippocampal contribution to contextual encoding is impaired by the extreme stress levels of trauma, producing a memory that is powerfully present but poorly located in time and place. The result is a memory that the brain cannot easily classify as finished, as something that happened then rather than something that is happening now, because the neural representation does not carry the contextual markers that ordinarily signal pastness. The trauma memory does not know it is over. The brain that holds it cannot find the frame that would tell it so. Directed forgetting, the deliberate attempt to suppress or not encode specific information, interacts with sleep in ways that research has only recently begun to examine. Studies on intentional memory suppression have found that the act of trying not to remember something requires active suppression of the hippocampal retrieval process. The same right dorsolateral prefrontal cortex mechanisms that suppress intrusive thoughts more generally. Research examining what happens to deliberately suppressed memories during sleep has found a complex picture. Material that was successfully suppressed during waking shows reduced overnight consolidation, suggesting that the suppression may partially prevent the memory from receiving the hippocampal replay it would otherwise get. But the suppression is not complete. Residual neural traces of the suppressed material appear to persist and can surface in the hypnagogic state and in dreams, where prefrontal suppression is significantly weakened and the hippocampus is less subject to top-down control. The material that was told not to be remembered tends to find the night a more welcoming environment than the day. The sleeping brain is less obedient to instructions about what should not be thought. It tends to think it anyway. One of the most quietly unsettling findings in memory research concerns the brain's apparent inability, under ordinary conditions, to reliably distinguish between a memory of something that actually happened and a memory of something vividly imagined. Research on reality monitoring, the process by which the brain assigns memories to the category of real or imagined experience, has found that this process depends on contextual cues at encoding, the perceptual richness, the sensory detail, the emotional tone, and the narrative context of the original experience. When a vivid imagination closely resembles these features of real experience, the subsequent memory of it can be stored with the same neural signature as a genuine episodic memory, and retrieved, sometimes even in the face of contradictory evidence, with full confidence in its authenticity. Research has found that this susceptibility to false memory formation increases under conditions of high emotional arousal, of sleep deprivation, and of repeated rehearsal of the imagined content, all of which can strengthen the neural representation of the imagined event to the point where it competes successfully with real memories for the status of what happened. The mind cannot always tell the difference between what it lived and what it only lived inside itself. Neither, sometimes, can the night. Sleep actively participates in the formation of false memories under specific conditions, a finding that has complicated the picture of sleep as purely memory protective. Research by Jessica Payne and colleagues, and by others examining how sleep transforms memories, has found that the abstracting and integrating function of overnight consolidation, which extracts general patterns from specific instances, can produce false recognition of new items that share the features of previously learned material, but were never actually encountered. Participants who slept between learning a set of related words and being tested on recognition were more likely to falsely recognize new words that were semantically related to the learned set than participants who remained awake. The sleep had done exactly what it was supposed to do, extracted the semantic structure, generalized across specific instances, and in doing so had created a convincing representation of something that never quite existed. Sleep makes the brain better at finding patterns. It also makes it better at finding patterns that are not entirely there. The role of prior knowledge and existing memory schemas in determining what new information survives. Overnight consolidation represents one of the most active areas of current memory research. Studies examining how new information is integrated with related prior knowledge have found that material that fits coherently into an existing knowledge structure, that connects to things the brain already knows, is consolidated more efficiently, consolidated faster, and retained more durably than comparable material that has no such connection. Research from the laboratory of Eleanor Maguire and Hugo Spears at University College London has found that the hippocampus appears to have a dedicated function in detecting when new information fits existing schemas and accelerating its integration. The practical implication is that the brain does not simply store new memories alongside old ones. It stores them into old ones, and the extent to which a new memory can find its place in what already exists determines, in significant part, how long it will remain retrievable. A new memory that has nowhere to connect to tends to be the first one the night releases. The overnight insight effect, the well-documented phenomenon in which sleep enables the sudden understanding of a solution or connection that the waking mind could not reach, represents the memory integrating function of sleep at its most practically visible. Research by Ulrich Wagner and colleagues at the University of Lübeck, published in Nature in 2004, demonstrated that participants who learned a mathematical task with a hidden shortcut were more than twice as likely to discover the shortcut after a night of sleep than after an equivalent waking interval. Even though neither group had been told a shortcut existed and none had consciously worked on the problem during the intervening period, the sleeping brain had apparently reorganized its representation of the learned material in a way that exposed the relationship the waking mind had encoded but not seen. Sleep did not simply preserve what was learned. It rearranged it until the arrangement revealed something that was always there. The insight did not come from sleeping on a problem. It came from what sleep did to the way the problem was represented. Procedural memory, the memory for skills, sequences, and learned motor patterns, consolidates during sleep through mechanisms that are partly distinct from those governing episodic and semantic memory. Research on motor skill learning and sleep has found that performance on a newly learned motor sequence, typing a specific pattern of finger movements, for example, improves significantly between an evening training session and a morning test that includes sleep, but shows no equivalent improvement over a waking interval of the same length. The consolidation appears to occur primarily during slow-wave sleep in the early part of the night and during REM sleep in the later part, with each contributing differently to different aspects of the learned skill. The accuracy improvements clustering in the slow-wave period and the speed improvements more associated with REM. The sleeping brain does not simply rest from learning a skill. It continues to practice the skill in its own way, reinforcing the neural sequence, smoothing the connections between steps, refining the representation until what was effortful becomes something closer to automatic. The night practices what the day learned. By morning, the body knows things the evening did not. The distinction between forgetting and retrieval failure, between a memory that is genuinely gone and one that is present but unreachable, is not simply philosophical. Research using techniques that probe the neural traces of memories that cannot be consciously retrieved has found consistent evidence that many memories classified as forgotten retain a detectable neural substrate, a weakened but present synaptic representation that can be accessed under the right conditions of cueing, context reinstatement, or physiological state. Studies using targeted memory reactivation to attempt retrieval of ostensibly forgotten material have found, in some paradigms, that cues delivered during sleep could strengthen memories that a post-sleep retrieval test would otherwise classify as lost, suggesting that the memory was present in the hippocampus but below the threshold of retrieval under standard test conditions. The forgotten memory and the unfindable memory look identical from the outside. From inside the neural substrate, they are different things. The brain that cannot find a memory has not necessarily lost it. It may simply not have the right key for the door it is behind. How memories that are never retrieved change over time. What happens to experiences that are stored but never visited again is a question that research has only partially answered. What is known is that unretrieved memories are not immune to change simply by virtue of being undisturbed. Research on the spontaneous decay of memory representations has found that neural traces of unaccessed memories show progressive weakening of their synaptic representations over time, and that memories subject to proactive and retroactive interference from similar material encoded before or after them show accelerated loss of discriminability even without being retrieved. The night processes memories regardless of whether the waking mind returns to them. The sleeping brain's replay and consolidation processes run on schedules determined by the memory's own encoding properties, not by the conscious mind's decision to revisit it. A memory left entirely alone is still touched by every night that passes over it. Every memory is subject to the night, whether or not the day ever thinks of it again. The relationship between sleep and the interference of similar memories. The well-documented phenomenon in which learning similar material before or after an original learning experience impairs retention of that original material has a specific and protective dimension. Research has found that sleep following learning significantly reduces the retroactive interference produced by subsequent similar learning, apparently by stabilizing the originally learned material's neural representation in a way that makes it more resistant to being disrupted by the new competing material. This is a practically important finding. The advice to sleep after studying, rather than immediately following study with additional learning in the same domain, has a specific neural rationale. The sleep seals the memory against what comes next. Without it, the new material moves into the same neural territory, and the original experience begins to lose its distinct address. Sleep builds a wall around what was just learned. Without it, the next thing learned tends to occupy the same ground. What is ultimately being described across all of these mechanisms of consolidation, selective forgetting, reconsolidation, schema integration, and overnight abstraction is not a filing system. It is something more dynamic and more personal. The brain's ongoing revision of the self's relationship to its own history. Each night, without the conscious mind's participation, decisions are made about what the past was, what it meant, and how it connects to what is known. The experiential specifics are gradually released while the relational structure, what connected to what mattered, what should be expected, is preserved and deepened. The person who wakes in the morning is not simply rested. They are, in a specific and measurable sense, slightly different in their relationship to everything they have ever experienced, because the night just finished revising that relationship in 1,000 small ways, none of them visible, all of them accumulating toward who the person is becoming. The night does not only keep the past, it slowly decides what the past was for. Something comes back that was not called. A face from a time the current life has almost no connection to. A room with a specific quality of light, a feeling that belongs to someone the current self barely recognizes as having been. These arrivals are not accidents. They are the surface expression of a very specific set of neural events, events that have been running quietly and that the night has had a hand in all along. Involuntary autobiographical memories, memories that surface without any deliberate retrieval attempt, arriving unbidden into awareness, have a neural basis that research published in behavioral and brain sciences has proposed to explain through spontaneous neural replay in the default mode network and hippocampus. The proposal developed from the observation that involuntary memories and voluntary ones appear to share the same fundamental retrieval mechanism of hippocampal pattern completion, locates the difference not in the machinery but in its initiation. Voluntary memories are triggered by a deliberate search process, while involuntary memories arise when the default mode network's ongoing associative activity spontaneously activates a hippocampal pattern that reaches the threshold for conscious retrieval without being directed there. The default mode network, running its continuous background process of self-referential association during any period of reduced external demand, effectively searches the autobiographical archive without being asked, and occasionally finds something. The uninvited memory was found by a search the mind was running without knowing it had started one. The neural signature of involuntary autobiographical memory retrieval has been measured in EEG and fMRI studies, comparing it to voluntary recall. Research has found that involuntary memories are associated with a faster and more complete initial activation of the memory representation. The hippocampal pattern completion that underlies retrieval occurs more rapidly and with less prefrontal involvement than voluntary recall, consistent with a process in which the memory arrives before the executive mind has had the opportunity to manage or shape what is retrieved. This speed and completeness is part of what gives involuntary memories their characteristic phenomenological quality, the sense not of remembering but of the past being suddenly present, arriving whole rather than being assembled piece by piece. The voluntary memory is built. The involuntary memory arrives already built. The memory that was not called comes fully formed. The one that was called tends to arrive in pieces. Déjà vu, the unsettling experience of feeling that a present moment has been experienced before, shares a neural substrate with involuntary autobiographical memory in ways that recent research has begun to clarify. The proposal from the same framework is that déjà vu involves a transient, spontaneous activation of the default mode network's familiarity detection circuitry without a corresponding full hippocampal retrieval event. The sense of recognition fires, but the specific memory that would explain it does not surface. This is the mirror image of involuntary memory. Instead of a past experience arriving without being called, the feeling that an experience belongs to the past arrives without the experience that would justify it. Research on the timing and contexts of déjà vu has found that it is more frequent in states of fatigue and during transitions between sleep and waking, conditions in which the boundary between the default mode network's internal activity and conscious awareness is less well defined. Déjà vu is the feeling of a memory without the memory itself. The archive signaled. The record did not come through. The reminiscence bump, the well-documented tendency for adults over 40 to retrieve a disproportionate number of autobiographical memories from the period between approximately 10 and 30 years of age, is one of the most consistently replicated findings in the study of memory across the lifespan. Research on the reminiscence bump, reviewed in a systematic analysis published in PMC, has identified at least two partially overlapping explanations for the effect. The identity formation account proposes that memories from adolescence and early adulthood are encoded with particular depth and durability because they are acquired during the period when the self is most actively being constructed. When the answer to the question of who one is remains genuinely open, and experiences are consequently enrolled in the larger project of self-definition in ways that give them organizational significance beyond their specific content. The cognitive account adds that this period tends to produce more novel first-time experiences, first love, first independence, first encounters with the full complexity of the adult world, and that novelty is one of the strongest predictors of durable encoding because the brain allocates more resources to experiences for which no prior schema exists. The memories that survive longest are often the ones from the time when everything was happening for the first time. The identity formation account of the reminiscence bump has specific implications for why memories from this period are disproportionately likely to surface in the pre-sleep hypnagogic state and in spontaneous memory retrieval across the lifespan. Research on self-defining memories, a concept developed by Jefferson Singer and colleagues to describe the memories that people identifies as most central to their sense of who they are, has found that these memories are more emotionally specific, more narratively elaborated, and more frequently rehearsed than other autobiographical memories of equivalent age. Rehearsal, the repeated retrieval and re-encoding of a memory, is one of the most reliable predictors of long-term retention, and the memories from the reminiscence bump period are precisely those that have been most actively recruited into the ongoing project of self-narration across decades. The night that replays these memories is not doing something special with them. It is continuing a process that the waking mind began long ago and has never stopped. The adolescent memories are not simply older. They have been used more, enrolled in the story of the self so many times that the path to them is wider than the path to almost anything else. the musical reminiscence bump. The finding that songs encountered during adolescence retain a disproportionate emotional and mnemonic salience across the lifespan has a specific neural explanation that research on adolescent brain development has recently elaborated. Research published in Frontiers in Psychology and reviewed in Neuroscience News has found that the dopaminergic reward system is at peak developmental sensitivity during adolescence. The mesolimbic dopamine pathways that assign emotional and motivational salience to experience show heightened reactivity during this period, meaning that the experiences of adolescence, including musical ones, are encoded with a level of reward circuit participation that most later experiences do not receive. Music encountered during this period is not simply remembered. It is encoded with a tag from the reward system that persists across decades and makes the song, when encountered again, a retrieval cue for the entire emotional context in which it was first heard. The song at the edge of memory is not simply a song. It is the year and the room and the person who was listening. The songs from adolescence does not bring back a memory. It brings back the person who was present when the memory was made. Spatial memory and episodic memory, the memory for places and the memory for events, share a common hippocampal infrastructure that research on place cells, grid cells, and episodic encoding has progressively revealed to be not incidental but architecturally fundamental. The same hippocampal circuitry that John O'Keefe, May-Britt Moser, and Edvard Moser identified as providing the brain's internal spatial mapping system, work for which they received the Nobel Prize in Physiology or Medicine in 2014, is the circuitry that supports the encoding and retrieval of episodic memories more generally. Research has found that episodic memories are organized around a spatial scaffold. The location in which an experience occurred provides part of the retrieval structure for the experience itself, which means that physically returning to a place where something significant happened is one of the most reliable triggers for the involuntary associated with memories, associated with that place. The brain that walks back into a childhood room does not simply recognize it. It finds, distributed through the geometry of the space, a set of retrieval cues for everything that occurred within it. The place is not where the memory happened. It is part of how the memory was stored. Walking back in is a search query. Remote memory consolidation. The decades-long process by which memories become progressively less dependent on the hippocampus and more distributed across neocortical networks has been a subject of theoretical debate between two competing models. The standard model of systems consolidation proposes that memories eventually become entirely hippocampus independent, their representation fully transferred to the neocortex. The multiple trace theory, proposed by Lynn Nadel and Morris Moscovich, argues that the hippocampus remains engaged in the retrieval of detailed, contextually rich episodic memories indefinitely, with only their more schematic, generalized aspects transferring to neocortex-only representations. Research published in Nature in 2026 examining the hippocampal and neocortical contributions to remote fear memory has found evidence consistent with a continuing interdependence. The hippocampus and anterior cingulate cortex interact dynamically during remote memory retrieval, with hippocampal ACC oscillatory coupling during sleep remaining important for remote memory maintenance long after the initial consolidation period. The most detailed and contextually specific old memories may retain their hippocampal connection, not because the consolidation process failed, but because the richness of episodic detail requires it. Some memories never fully leave the hippocampus. They are too specific, too contextual, too full of exactly how it felt to survive only as a general outline. The transformation of a memory's neural representation across decades is not simply a matter of moving from one location to another. Research on trace transformation theory, an extension of systems consolidation accounts, has found that what changes during remote memory consolidation is not only the neural address of the memory, but its content and structure. The episodic specifics that characterized the original encoding are progressively replaced by a more schematic, semantically organized representation that preserves the relational structure and emotional core of the experience while losing its perceptual and contextual detail. This is why very old memories often feel both profoundly real and strangely incomplete. The emotional conviction of the memory, its sense of personal significance, is intact, while the specific sensory details have thinned to outlines. Research has found that this transformation is not experienced as loss from the inside. The person does not feel that the memory has become less complete. The schema that remains feels, subjectively, like the whole thing. What survives decades is the meaning of the experience. The experience itself, in its original fullness, was let go long ago, so gradually that the letting go was never felt. The self-reference effect in memory. The well-replicated finding that information processed in relation to the self is remembered significantly better than equivalent information processed in relation to other people or in relation to abstract categories has a sleep-related dimension that research has only recently examined. Studies on the self-reference effect and overnight consolidation have found that self-referentially encoded material shows preferentially enhanced consolidation during sleep compared to semantically equivalent material encoded without self-reference, as self-relevant at encoding. Influences not only its initial depth of processing, but its priority during a overnight memory transfer. The self that sleeps is in part curating itself. The overnight processes that determine what is strengthened and what is released are responsive to what the self considered personally relevant, and the memories that survive the night in best condition are disproportionately the ones the person thought, consciously or not, were about them. The brain keeps what it considers most likely to be needed by whoever it is keeping it for. The positivity effect in aging memory. The finding that older adults show a relative preference for encoding and retrieving positive emotional material over negative material compared to younger adults who show the opposite pattern has a consolidation-related mechanism that research on emotional memory and aging has proposed. Studies have found that the attentional and encoding shift toward positive material in older adults is associated with preserved amylactivity to positive stimuli alongside reduced reactivity to negative ones. A selective change that appears to reflect both motivational and neurobiological shifts in how the aging brain assigns emotional priority. The overnight consolidation processes that preferentially strengthen emotionally tagged material, therefore, in the aging brain, preferentially strengthen positive memories. The result, accumulated across years of sleep, is an autobiographical archive that becomes progressively weighted toward what went well, not through deliberate revision, but through the nightly arithmetic of what the brain decided was worth keeping. The older memory is not merely old. It has been filtered by decades of nights that progressively kept more of the warmth. What the morning retrieves that the evening could not is one of the most practically observable expressions of what the night does to memory. Research on the timing of memory retrieval relative to sleep has consistently found that information tested immediately after learning and then retested the following morning shows measurably better performance on the morning test, not because the interval alone allowed forgetting to stabilize, but because the specific processes of overnight consolidation have strengthened and reorganized the memory's neural representation in ways that make it more accessible and more precisely retrievable. This improvement is not uniform across all memory types, procedural skills, emotional memories, and memories that require connecting separate pieces of information show among the largest overnight gains. The mind that went to sleep, unable to solve something, retrieve something, or understand how two things fit together, sometimes wakes with the answer present, not as a vague intuition, but as a specific, retrievable, immediately usable piece of knowledge that the night quietly finished building. The morning knows things the evening did not. The night worked while the mind was away from it, the incubation effect. The well-documented phenomenon in which a period of time away from a problem, particularly a period that includes sleep, increases the probability of sudden insight, has a memory-based explanation that research has largely converged on. The proposed mechanism involves the loosening of inappropriate mental sets. During waking, the initial framing of a problem tends to activate and repeatedly reinforce the memories and associations most closely related to that framing, creating a retrieval rut that constrains what the mind can see. During sleep, particularly during REM sleep's low norepinephrine associative state, the constraints that structured the waking approach relax, and the memory system is free to activate connections that the waking mind's framing had been suppressing. The insight arrives in the morning, not because the problem was solved during sleep, but because sleep changed the memory landscape in which the problem is being approached. The same information, now connected differently, makes something visible that was not visible before. The problem did not change overnight. The memory of everything connected to it did. That is what made the answer findable. The social dimension of memory, the way in which telling a story about a personal experience changes the memory of that experience, has implications for what the night processes when the day has included storytelling. Research on the misinformation effect and social contagion of memory has found that describing an experience to another person produces a version of the experience that is shaped by the demands of the telling, compressed, narratively coherent, simplified in ways that a conversation requires. This told version competes with the original encoding during subsequent retrieval, and research has found that multiple retellings progressively shift the accessible version of the memory toward the narrative that was rehearsed socially rather than the raw episode that was originally encoded. The night that follows a day of storytelling consolidates, among other things, the stories, the publicly shaped versions of the self's history that the day produced. What is strengthened is not always what happened. Sometimes it is the version that was told about what happened. The memory that is shared becomes, over time, the memory that is kept. The untold version fades into what the told version replaced. The self as a narrative, the understanding of personal identity as an ongoing story that the mind constructs from what it retrieves, has a memory science foundation that makes the night's work directly constitutive of who a person is. Research by Dan McAdams on narrative identity and by Martin Conway on the working self-concept and autobiographical memory has found that the self is not a stable object that memory serves. It is continuously assembled from the memories that are currently most accessible, shaped by the emotional tone of what is retrieved and revised with each new night's consolidation decisions. The person whose recent nights have strengthened positive, self-affirming memories is, in a specific and measurable sense, living in a different autobiographical landscape than the person whose consolidation has been shaped by stress, loss, or disrupted sleep. Not because their histories are different, but because the accessible representation of those histories has been shaped by different nightly processes. The self is not remembered. It is built incrementally and continuously from what the night decides to make retrievable identity is not what the person has been. It is what the sleeping brain has decided to keep bringing back. There is something the night has been doing all along that the previous parts have approached, but not yet named directly. Not the mechanism of it, the oscillations, the replay, the transfer, but what all of it adds up to, taken together across a lifetime of nights. What it means that the brain keeps what it keeps. What it means that some things come back and some things do not. What it means that the self wakes each morning shaped by decisions made in the dark. Flashbulb memories, the unusually vivid, detailed, and long-lasting memories that form around events of high emotional significance and personal consequence, represent the extreme end of a continuum that reveals what the memory system is ultimately optimizing for. Research on flashbulb memories, developed through work by Roger Brown and James Kulik, and extended by subsequent investigators, has found that these memories are encoded and consolidated with a completeness and durability that ordinary memories rarely achieve. Not because the brain has a special recording mode, but because the neurochemical conditions of their encoding are exceptional. The combination of intense amygdala activation, high norepinephrine, elevated cortisol, and the social rehearsal that typically follows a major event produces a convergence of consolidation-promoting factors that makes the resulting memory among the most robustly stored in the system. Research has found that flashbulb memories show elevated activity in the hippocampus, amygdala, and lateral prefrontal cortex during retrieval even decades after encoding. A pattern that reflects not photographic accuracy, but deeply consolidated significance. They are not remembered more faithfully than other memories. They are remembered more persistently. The most significant moments are not remembered more accurately. They are remembered with more force. The dreaming brain is, among other things, a memory laboratory, a space in which stored material is recombined, recontextualized, and tested against other stored material in combinations that waking cognition rarely attempts. Research on the relationship between dream content and autobiographical memory has found that dream narratives draw selectively from specific memory windows. Recent memories from the past day or two, older memories from emotionally significant periods, and fragments of both combined in ways that reflect the associative logic of the memory system rather than the causal logic of waking experience. Research by Aaron Walmsley and colleagues has found that dreaming about the material of a recently learned task significantly predicts the magnitude of overnight improvement on that task. The dream is not simply a reflection of consolidation, but may be a part of it, the brain's narrative faculty participating in the integration process by constructing scenarios that test the connections between what is newly learned and what is already known. The dream does not merely replay the day. It rehearses what the day might mean against everything else the brain has ever stored. What the hypnagogic state, the transitional period between waking and sleep, is doing with the day's unresolved material has a specific character that makes it one of the richest and most functionally significant periods of the entire sleep-wake cycle. Research on hypnagogic mentation has found that the content of hypnagogic imagery and thought is disproportionately drawn from the emotional and cognitive residue of the preceding waking period. The concerns, questions, and unfinished emotional processing of the day surface in the hypnagogic state in a form that is more associatively free and less constrained by narrative logic than waking thought. This is not simply because the mind is wandering. Research has proposed that the hypnagogic state may serve a specific preparatory function, presenting the most emotionally and cognitively unresolved material to the associative and consolidation systems of the subsequent sleep. At the moment, those systems are most primed to receive it. The last things the drifting mind visits before sleep may be precisely the things the night will work on most. What is still unfinished at the edge of sleep is first in line for what the night will do next. Somatic memory, the body's retention of the physical and physiological patterns associated with experience, operates alongside and partly independently of the declarative memory systems that are most associated with sleep consolidation, but it shares the night's work in ways research has begun to document. Research on procedural and motor memory consolidation during sleep has established that the body's learning, the way a skill refines itself overnight, the way a physical pattern becomes more automatic with each night of sleep after its acquisition, involves specific neural processes during slow wave and REM sleep that rehearse the motor sequences at a neural level without requiring any physical movement. But beyond the procedural, the body holds other things, the physiological signatures of significant experience, the pattern of arousal, the muscle tension, the autonomic state that were encoded alongside the declarative memory of the event. Research on the body-based dimensions of memory has found that these somatic traces are retrieved alongside the declarative memory they accompanied and contribute to the phenomenological completeness of the remembered experience. The memory of grief that lands in the chest, the memory of joy that arrives with something physical. The body remembers the feeling of the experience, not as a metaphor, but as a recovered physiological state, grief. The sustained emotional and cognitive processing that follows significant loss has a relationship with sleep and memory that research has found to be both neurologically specific and deeply meaningful. The task of grief, understood from a memory perspective, involves the gradual transformation of a living relationship into a memory. The shift from a present connection that shapes daily expectation and behavior to a past connection that is accessible through retrieval but no longer available in the present tense. Research on grief and sleep has found that this transformation depends substantially on the same overnight memory processing that consolidates any significant emotional material. REM sleep's emotional attenuation function progressively softens the acute pain attached to the retrieved memory of the lost person. Slow wave sleep's consolidation function preserves and deepens the memory's positive and meaningful content, and the ongoing nightly work of systems consolidation gradually moves the most important memories of the relationship toward the neocortical networks where they will remain accessible without requiring the acute hippocampal activation that characterizes fresh grief. Sleep does not end grief. It does the work, slowly and without being asked, that allows grief to become something the person can carry, rather than something that carries them. Every night of sleep after a loss is, quietly, part of learning how to hold the person who is gone. What happens when a forgotten memory returns? When something that felt genuinely gone surfaces, whole and specific, in the drift before sleep, or in the morning's first soft moments of awareness, has a phenomenological quality that research can describe neurologically, but that the experience itself exceeds. The neural event is a pattern completion, a partial cue reaching the hippocampus, activating a stored representation that crosses the threshold of conscious retrieval. But what the person experiences is something that feels more like discovery than retrieval. The memory arrives with a sense of its own weight, not only the content of what happened, but the texture of what it was to be the person it happened to. At that time, in that place, with whatever was then still unknown about what would come next. Research on the phenomenology of involuntary memory retrieval has consistently found that these returns are experienced as more emotionally intense, more temporally disorienting, and more personally significant than voluntary recall of equivalent material. The forgotten memory does not simply return, it returns with the self that lived it. The memory comes back with the person inside it. Both were gone, both returned together. Musical memory is the last cognitive capacity to be significantly impaired in Alzheimer's disease and related dementias, a finding that has been replicated across multiple research groups and has specific neural implications. Research on musical memory and neurodegeneration has found that the brain regions most critically involved in musical memory, including areas of the medial prefrontal cortex associated with personally meaningful music and the cerebellum involved in rhythmic processing, are among the last to be affected by the neurofibrillary tangles and amyloid plaques that characterize Alzheimer's pathology. The hippocampus, which is among the earliest and most severely affected regions, is less critical to the retrieval of long-established musical memories than to the retrieval of other autobiographical material, consistent with the remote consolidation model in which well-rehearsed, emotionally and self-referentially encoded material achieves the most hippocampus-independent representation over time. A person who cannot remember their own name may still recognize and be moved by the song that was playing when something important happened to them 50 years ago. Music reaches past the damage. It was stored somewhere the disease arrives last. The hippocampus does not only manage the past. Research on the hippocampus's role in future thinking, the simulation of anticipated events, has found that the same neural circuitry that retrieves episodic memories is recruited when imagining future scenarios, to the extent that patients with hippocampal damage show specific impairment not only in retrieving the past, but in constructing coherent imagined futures. The proposed mechanism, developed by Demis Hassabis, Eleanor Maguire, and colleagues, suggests that episodic memory is the raw material from which the brain constructs mental simulations of what might happen next. The episodic future, thought that underlies planning, anticipation, and imagination. The memory the night consolidates is therefore not only a record of what was, it is the material from which the waking mind will build its models of what might be. What is kept from the past shapes what can be imagined about the future. The past that is preserved tonight becomes the future that can be conceived tomorrow. The persistent memories, those that do not decay significantly across decades, share a cluster of properties that research has identified with unusual consistency. They are emotionally significant. They are novel. Among the first times an experience of their type occurred, they are self-relevant, enrolled in the narrative of who the person is. They have been rehearsed either socially or internally, enough times that the retrieval path is well worn, and they were consolidated on a night of good sleep within a window close enough to their encoding that the hippocampal representation was still rich when the replay began. Research on very long-term memory retention has found that memories satisfying all of these conditions can remain retrievable with remarkable fidelity across an entire lifetime. Not unchanged, because every retrieval revises slightly but recognizable, specific, and emotionally present in ways that most memories of equivalent age are not. The brain does not preserve equally, it preserves deliberately, according to a logic that the conscious mind rarely sees clearly, but that the self experiences as simply. These are the things I have not forgotten. The memories kept for a lifetime were not simply lucky. They were built to last by conditions present at their beginning. What all of this research ultimately converges on is a picture of identity that is more fragile and more resilient than either characterization alone would suggest. Fragile, because the memories that constitute the self are reconstructive rather than reproductive, are subject to modification with each retrieval, are shaped by the emotional conditions under which they are consolidated, and are dependent on nights of sleep that life does not always provide. Resilient, because the system that manages them is redundant, adaptive, and oriented toward preservation, toward keeping what matters, softening what hurts, connecting what was separate, and returning on the right morning something that felt entirely gone. The self is not a fixed object that memory serves. It is the ongoing outcome of what the night decides to do with the day, and it has survived, so far, every night it has been given. What the brain keeps is what the self is made of. The night has been doing this work since the beginning. There is something small at the center of all of this, something that the neuroscience describes but does not quite reach. Every night, in the quiet after the day has finished, the brain goes back through what happened. Not all of it, the parts that mattered. It replays them at speeds the conscious mind will never perceive. It moves them carefully to places they can last. It softens some of them. It connects others to things they had never been connected to before. It releases what is no longer needed. And it does all of this while the person who lived the day is somewhere else entirely, in a dream or in nothing, or in the slow drift between. The work is invisible. The outcome is the person who wakes in the morning, slightly different from the one who lay down, carrying a past that the night has quietly been tending all along. The night has been taking care of what you lived. It does this every time. It has never stopped. If this kept you company tonight, please like and subscribe.